Hello, my name is Irene Pham, and today I will be presenting my research on the influence of bacterial inoculum density effect on host fitness. The mutualistic symbiotic relationship between host and bacteria has been passed down vertically from mother to offspring and has been essential to the well-being of the host. However, this is not the case for several families of choroid and lygoid bugs as they acquire their symbiont from their environment. The clade of Caliborronia has been found in the amphor gut region of Leptoglossus phyllops. Leptoglossus phyllops is a common polyphagous agricultural pest found in North and South America that feeds on tomatoes and citrus. They acquire their symbiont primarily during their second developmental stage. After the symbiont is acquired by the bug, the bacteria grows to a high abundance and the excess bacteria are then digested by the bugs to absorb the necessary amino acids and B vitamins. The purpose of this research is to answer the question, does the density of Caliborronia affect the fitness of Leptoglossus phyllops? We hypothesized that if the Leptoglossus phyllops were given a low density of Caliborronia, then their fitness would be worse in comparison to Leptoglossus phyllops that were given a higher density of bacteria. The experiment was done by inoculating Leptoglossus phyllops with the isolate Lep1A1, a commonly isolated Caliborronia strain native to Leptoglossus phyllops found in Texas. This strain was chosen because previous studies have shown Lep1A1 to be one of the best colonizing strains within the bug. The strain were cultured and serially diluted to 3 different concentrations. The concentrations were 1 × 10 to the 4 cell per milliliter, 1 × 10 to the 6 cell per milliliter, and 2 × 10 to the 7 cell per milliliter. There were 9 trials replicated, with each trial containing 8 to 10 bugs. After inoculating the bugs for 24 hours, the bugs' development time were tracked, and once they had reached adulthood, the mass was measured for each concentration. For the results, we measured the time it took them to reach development between their 3rd developmental stage to their adulthood and their mass once they reach adulthood, as previous research had indicated that it was a good indicator of the bug's overall fitness. The data show that there were no significant differences between the bug's mass and the development of the insect when infected with different density inoculation. The p-value for the effect of the inoculant density on the host mass and development was both very high. This means that the host did not exhibit higher fitness when infected with a higher dosage of symbiont. This means that our hypothesis was not supported. An explanation as to why there is no correlation between the inoculum density and host fitness. It seems that regardless of the initial amount of bacteria that the bugs were exposed to, the bacteria grows to sufficient density inside the gut that is needed for proper growth and development. And after the bacteria has grown to the desired threshold, the bug would then digest the excess bacteria to absorb the necessary amino acids and B vitamins. As the global population continued to rise globally and the need for more food increases, the use of pesticides, which harms the environment, has also increased. It is important to find an alternative method to increase crop yield without the use of pesticide. Determining the initial Caliborronia infection density at which bug fitness starts to decline may provide an alternative method of controlling pest control. Since the concentration that we had used was not low enough, future research would involve further lowering the inoculation dosage to determine the minimum amount of bacteria that the Leptoglossus phyllops would need to get inoculated. Additionally, studies would need to be conducted to determine the amount of live Caliborronia that can be found in soil when Leptoglossus are present versus when they are not. I would like to thank Dr. Allison Ravenscraft and Bibek Parajuli for their mentor and guidance on this research. I would also like to additionally thank my lab mate for their additional lab support.